Thanks for tuning in to BEaTS Research Radio, a podcast and YouTube channel dedicated to bringing our community closer to biomedical research in the science and engineering fields. I'm Hallie Arnott, a biomedical engineering master's student at the University of Ottawa, and I'll be your host on today's episode. Joining us is Dr. Benjamin Hibbert, an interventional cardiologist and director of the Vascular Biology and Experimental Medicine Laboratory at the University of Ottawa Heart Institute. He's also an assistant professor in the Department of Medicine at the University of Ottawa. Dr. Hibbert's interests include both clinical and basic science research projects with the goal of translational research. His research has been performed clinically in patients undergoing percutaneous coronary interventions or structural heart disease procedures. Today, Dr. Hibbert is here to speak with us about the importance of preclinical studies for translational research. Thank you so much for joining us here today on BEaTS Research Radio.
Thank you very much for the invitation.
For my first question, I'd like to ask you a little bit about being a clinician scientist. As a clinician scientist, you are immersed in both the research side of medicine as well as the clinical side. Can you tell us about the importance of translational medicine and how your research is engaging in this space?
Yeah, no, that's a great question. So I think true clinician scientists are a bit of a rare breed. And I'm certainly hoping by training some of the clinicians that are coming along and working with me that we get more and more people interested in research. I think the real advantage of having clinician scientists who do this kind of work is that you get a real application of the problems for treating your patients, and you identify gaps or needs in clinical care. Before I started med school, I used to do a tremendous amount of research at what you could consider the preclinical or translational stage. And often I didn't quite understand the context of the diseases that I was studying or trying to treat. By being a clinician and looking after patients day to day, you really get to identify where those needs are. And that helps focus the research. When we go back to the lab and we talk about problems that we're trying to solve, we know which problems we have on a day-to-day basis in terms of treating patients. And so that really enables us to focus the research. And so I think as a clinician scientist, that gives us a bit of a unique perspective. Of course, it presents with challenges. I was late for this interview because we were resuscitating a patient. This weekend, I was on call all weekend long. And so there does become competing time for your attention. And obviously, being a busy physician or clinician requires a lot of your time, both to look after the patient, stay up to date with the latest and greatest in medical care. And so that absolutely takes away from your research time. And I think that's where partnerships with researchers are also critically important for clinician scientists. So while I think there is advantages to being a clinician scientist, I do think it's important both to partner with pure clinicians, pure researchers, to really help focus that translation. I also find being a clinician scientist really motivating in the sense, as I had mentioned, identifying gaps in care, but, there's a real application of the work that we do. I look after patients who either suffer symptoms or morbidity from the disease process that they have, or in some cases, they pass away from the disease process that we treat. So it does also help keep me motivated and hits a little bit close to home in terms of really trying to develop those novel therapies so you can help those people that you're dealing with every day.
Definitely, I can see how the collaboration as well is an extremely important factor in there. Would you say, coming from the clinician scientist point of view, would you say your research mostly comes from the bench and then works its way towards the clinic, or do you sometimes get ideas in the reverse order where you see something in the clinic and then you try and work your way back to the basic science point of view?
Yeah, so that's a fascinating question, and you can delve into how the research enterprise develops. I really believe firmly that we do most of our science incorrectly, and by that I mean people become experts in a topic or a technique, and then they turn around and they try to apply that topic or that technique to different disease processes, whether or not it's relevant. There's a process called biodesign, and you can actually do master's degrees in there, and there are programs in this where you take an opposite approach. You go to the clinic, you go to the bedside, and you identify gaps in care, you identify unmet needs. And then you turn around and you say, oh, how do we address that unmet need? and you look for partners or experts that have the skill set to fill that back in. And it's very different than how we train scientists. It's very different than how we train clinician scientists. But I think that that's a much more efficient way to do focused research. And so I think it's really important, I think, that we go to the bedside first and say, where is that need? Finding a very complex or elegant solution to a problem that doesn't exist just means that we're never going to apply that to patient care. And that's the cold, hard reality of the practicality of looking after patients. Whereas if you identify an unmet need up front, and then you turn around and you laser focus your attention and your skill set on that problem, you're much more likely to make an impact on patient care. And it's certainly not how the scientific enterprise works. I've talked about this a lot in some of our research papers and some of the interviews like this that I've given. Like I said, I often find, and this was my experience during my research training, is people become experts in specific problems, and they don't really understand the clinical context that they're working in. So I really think that, and I do think that's where clinician scientists are particularly helpful for identifying those gaps. And so I like to think that a lot of the way I've approached a lot of my problems is first with the patient, identify that problem, and then turn around and look at what solutions we have available to us and how do we solve this problem. And more often than not, I'm not an expert in the skill set that I need to solve that problem. And so this is where collaborations and finding those people who are experts to solve that problem is critically important. In the last 2 weeks, I can think of multiple examples of that. So for example, one of the things I do is look at arteries and try to identify arteries and plaques that are vulnerable to cause a heart attack. And one of the things we thought about is, man, wouldn't it be awesome to have markers that identify these high-risk plaques? Well, I'm not an expert at building markers. And so we've partnered with basic scientists and translational scientists who are experts at building those markers. And then we'll go and test it in the large animals in patients and see if we can identify a problem. Similar, I'm not an expert in complex imaging. I do a lot of structural heart disease, and we've developed new tools or new devices to treat patients with. Well, we need those expertise in streamlining those processes. So I've partnered with someone who is an expert in artificial intelligence, machine learning, imaging, and brought them into the project to help solve that problem that we have clinically. So I do think that if your goal is to change patient care and to impact patient outcomes, you got to start with what's the problem first, and then work backwards to potential solutions.
Right. Yes, I can kind of visualize it as a reverse hierarchical structure with the patient being the final, but the initial idea there. So do you think that kind of among all of the reasons you talked about and having science be kind of going in the wrong direction currently from your point of view, do clinical trials, since most or many show like minor and neutral effects, even with large patient groups? So do you think that basic science and preclinical research can improve this?
Yeah. So again, that's a loaded question. So negative trials are super important. And I don't necessarily think that they represent failure of the scientific process. Hopefully, if we are taking risks and trying new things, most things should fail. And I don't think that that's necessarily a failure of the scientific process. I think publishing those studies is super important. I think doing those studies is super important. And that's how we learn. If a lab or a group is only publishing clinical trials, makes me a little bit suspicious about how they're doing their science and what they're doing, because the reality is most things shouldn't work. If you're doing truly novel or innovative things, that's not uncommon that things shouldn't work. That being said, what I worry about is if preclinical science isn't done well, there's two large problems that arise. Probably the most concerning is the one where a therapy that actually works, gets turned down or rejected because the science that was done initially just simply wasn't done well. And so we just never test that therapy. That worries me a lot because those studies just never get done. The studies that get done and end up being negative because the preclinical work wasn't really strong, well, there's a different problem with that. At least the science or the clinical study was done to test it. there's an element of, did you put patients at risk? Have you wasted resources? Have you wasted time? Because the fundamental work or the base of that work wasn't really strong. And that's true to all of those things. But, I would argue a little bit the system worked there where you tested it. Can the research enterprise be more efficient? So the reality is we all live, at a time of fiscal constraint. Those budgets are just going to get tighter with the current fiscal environment. So the question the question that I wonder about is, can we make the research enterprise more efficient? By being more collaborative, doing better and less science that really answers the question that we're trying to answer, so that the dollars that the public is giving us to do our work is most efficiently used. And I think that would restore a lot of faith in the scientific process. I think science is going to have a bit of a honeymoon period here because everyone's going to be super impressed with all the work that's happened with COVID. But you still wonder if the enterprise of science could be done more efficiently.
Right. And you recently did a systematic review, or were involved in a systematic review on atrial fibrillation-oriented therapies and I guess the preclinical research involved in that. Can you tell us about what the motivation was to look there and do that study, and what knowledge gap was addressed in the review?
Yeah, so that's actually work that was done by Dr. Daryl Davis and his lab. So they came to us for technical expertise. One of the areas I'm particularly interested in is systematic reviews and preclinical reviews of data. And again, this kind of stems from a process that I've learned in clinical medicine. When we go to do a clinical trial, we do a systematic review and evaluate the state of the evidence before we embark on a clinical trial. So we get all the trials that have been done, we evaluate all the evidence, we looked at what worked and what didn't work, and then we make the argument about whether or not we should move forward. You can certainly do those kinds of studies as Dr. Davis and his lab did with atrial fibrillation, and I think I think it's prudent to do that before you embark on researching a new strategy or a new therapeutic avenue, and you're testing it preclinically. You should do that type of analysis to see whether or not it's worthwhile, because your study is going to stand in the context of all these other studies. So having a solid foundation before you embark on testing this, I think, is really important. And they did a great job on that analysis. And I think it stands as a testament to the type of work that should be done before you embark on that. Certainly when I wear my clinical research hat, we do that every single time. So when a resident or a physician that's working with me wants to do a clinical trial, the first thing we do is a systematic review and a meta-analysis to say, what is the state of the evidence? What do we know already before we embark on this? Because nothing's worse than just repeating something that's already done, or worse, trying to refute a question that your study simply won't be powered to refute. And so I think that process, that iterative process of evaluating the state of the evidence, and then when you do your study, putting your study in the context of all that evidence is really important to help guide us. And it's not often done preclinically. I think that's gaining a little bit more momentum now. And I think we're going to see more and more of those types of studies. There's a lot of narrative reviews in preclinical or translational science, which is kind of like, here's my state of the art and my opinion on what's going on. Whereas a systematic review and a meta-analysis is a much more rigorous, much more objective type of analysis to kind of state where the science is, and enables the readers of those studies to really get a sense of where the data is, not what someone's opinion is of the data.
Right, absolutely. I think that that type of analysis is also just another form of collaboration. Whether or not you're working with that person, bringing everything together is still collaborative at that point. Would you say that going through that systematic review process, is it easy or not easy? It's definitely not easy, but is it, when you're looking at all of these preclinical studies and basic science research, does it seem to be, are the results from each of those studies presented in a way consistently? Are the results presented consistently? Are the same factors always presented to make that review possible? And because sometimes you can have things that aren't reported but maybe should have been. Is that common to find when you're doing that type of review?
Yeah, Yeah, it's a major problem both with clinical and with preclinical studies. If the data is not reported in the trial, they often get, or the study, they often just get excluded from this type of analysis. And it's definitely a bigger problem in the kind of the preclinical studies, because the outputs or the readouts are always different. You don't necessarily always report, the results of a proliferation assay the same way. A lot of the assays are homemade. And so not only are the point estimates different, but the variance difference within those assays. And so it's definitely more challenging than, say when we do a meta-analysis in clinical medicine, when we look at dead patients, are they dead or are they alive? That's very binary. And there's obviously no interpretation of those results. And so it's definitely more challenging in preclinical science. But what you can get is a good sense of the direction of all of these studies. So you get kind of a sense of, and you get a graphical representation, and you get point estimates of the relative effect sizes. And that enables you to look at it and say, wow, most of these studies are generally positive. They generally have a 20 to 30% effect size. This is probably what we could guess. And it's really important for powering studies. I gave a lecture yesterday on this. When we're doing preclinical science, it's really important to make sure that we're doing adequately powered studies, that the statistical foundation of the studies is rigorous. And that's also often not done, mostly because people just don't have a good sense of how much of an effect size should they expect. What kind of variance are they looking in the assay? And so those preclinical analyses, systematic reviews and meta-analyses certainly gives you a good sense of that. What I worry most about preclinical science is negative studies don't get published. And that is a plague on the field. And so, I'll give you an example. When I did EPC biology for my PhD with Ed O'Brien, who's in Calgary. And, one of the, kind of debates in the field is, could we get circulating peripheral blood cells to turn into smooth muscle cells? And there was a paper in Circulation that said that this was a thing. And, I spent several summers and thousands of dollars trying to replicate these studies and simply cannot. I got in contact with the authors, got the reagents from the same place and was never able to replicate it. And so, a huge amount of time and effort went into it. And it was funny when I went to conferences and talked to other labs that were working in the field and other groups, everyone had spent time trying to replicate that study. And, we all kind of just agreed that it just wasn't something that could be done. But we had all gone through that process of publishing it. Well, there really isn't a forum to say, look, you've published this paper in Circulation and I've tried to replicate it and I can't. There isn't a really, there's PubPeer and there's some things where that's starting to become more common. But man, you would hope that for the sake of efficiency of science that science is supposed to be self-correcting, that this would be more readily accepted. And so what I worry about when we do our systematic reviews at a preclinical level is that I worry that a lot of the negative studies just aren't there. And so we get a biased effect size to the positive. And so that certainly is a limitation.
Yes, I can see that being we could probably spend another hour talking about how important it is to publish negative results. But I'd also like to ask something about sex and gender bias in these studies. Do you find that a lot of the preclinical studies are in animals or using cells from animals? Would you say that it's disproportionate in terms of male, female? And is it reported every time which male or female type of animal is being used?
Yeah, so that was, Dan Ramirez, who's an electrophysiologist now in Ottawa, did his master's on that with me. And so we looked at sex bias in particular. We published those results in 2017 in Circulation, and it's shockingly bad. Everyone recognizes it as important, but only a fifth of studies actually do proper sex disaggregate analysis. The vast majority of studies use male only animals, and there are both practical reasons for that and historical reasons for that. I was taught when I went through many years ago when I was doing my research training, you use male animals. The estrous cycle, it's going to cause variability. We're not going to know where we are. That was just taught. There's also a practical aspect to it in the sense that, if you're breeding animals, you keep your females for breeding, you experiment on your males. And then often at the end, you simply destroy your females. You never experiment on them. And I think that's wrong for many reasons. We get a sex-biased view of disease patterns and response to therapies. So there are certainly therapies out there that work better in women than men, and if we test them only in male animals, we just think they don't work, and then of course our female patients don't get to benefit from that. Importantly, there's a difference between powering a study. So it's not that you have to replicate everything in a full cohort of men and a full cohort of women, but you have to at least report sex disaggregate data so that if there's a big discrepancy, you know about it. And then that might prompt you to look a little bit deeper. I always find it fascinating, particularly, we use mice so much in preclinical science. And there's a lot of limitations to that. Obviously, we can do genetic manipulations, and it's really improved our understanding of a lot of diseases. But we'll alter one gene, but then no one will ever check when you're doing your genotyping of your mice, do you get differences in males and females? You can alter one gene, and one cohort can be all males, one cohort can be all females. You have differences of an entire chromosome, and nobody seems to care about it as long as your one mutation is different between the groups. And so there's an incredible amount, I'm certain there's an incredible amount of reported scientific differences, which are owing to sex differences and not necessarily to the mutations that we've introduced, and people just simply don't check. I think journals recognize it now. The NIH certainly recognizes it now. We try to do a really good job of it, obviously, because it's something we're interested in, of always reporting sex disaggregate data. But it's funny, even when we submit some papers, some journals will put it in as a supplemental, and some journals will say to us, it's underpowered, don't report it. And to which we often reply, well, no, you have to report it. And in clinical trials, we always have to break down our data by male and female, and I think that's good. And I think that's just going to take time to change in the scientific culture. So do I think it's a problem? Absolutely. Do I think it's been adequately addressed? Absolutely not. I just think there's a lack of awareness of it. How do I think we fix it? Well, I think that journals should mandate that you, especially for animal studies, you always report sex disaggregate data. I think funding agencies should mandate it. And not at the level of, we all check the box on CHR, we're going to do sex-based analysis, but they should actually verify it so that if you go back for your next grant or your next funding opportunity, then you haven't reported that in your paper or you haven't addressed that. That should disqualify you from research or from getting funding. I think there's a lot of lip service that's paid to it. I think that, when journals mandate it for submission, when funding agencies mandate it for getting your grant, I think we'll see it addressed.
Right. Yes, that's also a very interesting topic, and just as you were saying, it should be recognized and looked at on a higher level, and to make sure that everyone is complying. So that is going to conclude our interview today. I'd like to thank you so much for joining us here on BEaTS Research Radio and talking to us about your translational research and the importance of preclinical studies for translational research.
Thank you very much for the invitation. Really appreciate it.
BEaTS Research Radio is supported by the University of Ottawa Heart Institute, the BEaTS Research Lab, and the Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa. Our director is Dr. Emilio Alarcon. Thank you so much for tuning in, and we'll catch you all next week.